In the prior videos, you saw Michelle Navard and myself talk about the theoretical motivation and background for genomic-SEM and LD score regression. In this next set of videos, I'm going to be going over the pragmatics of how the actual function, or the package, I should say, in genomic-SEM is run. Starting with step one, the munge function, where munge refers to a general reformatting of data, where here we're formatting the data to look like and be aligned across data sets in ways that is needed for LD score regression. Prior to running munge, a first step is identifying where you're going to get the data that goes into your statistical models. GWAS data is widely available at this point, and there's lots of different places you can go. A list, certainly not a comprehensive list, but a starting point is available on the genomic-SEM GitHub that includes different data resources. And just to highlight a few here, there's different biobanks, which reflect GWAS that were done within a particular data set. There's more general resources like GWAS catalog, where you can look to see if a GWAS has been done for a trait of interest across any data set. And then consortia that don't reflect a single data set, but instead have brought together multiple different cohorts to meta-analyze and produce well-powered GWAS for traits of interest, like psychiatric disorders for the PGC. When you're looking for GWAS data, there are 5 key pieces of information that you need in order to be able to run munge and LD score regression. The first is the RSID of the SNP, or the SNP identifier. The second is the A1 column, which is the affect allele. The third, the A2 column, or the non-affect allele. The fourth is a column that could be turned into a sign-effect column. So this could be a z-statistic, logistic, or continuous betas, or an odds ratio. The only issue would be if you only have p-values. But with that said, the fifth piece of required information is the p-value. So the best way to figure this out is simply to look at the README file for the GWAS data, or to open the first couple of rows of a data set that you're planning on using. So this is a data set that I'm going to use in the video for the PTSD GWAS that includes those 5 pieces of information: the RSID, allele 1 or A1, allele 2 or A2, the signed effect column, and the p-value. So this data set is suitable for running through munge. In this example, I'm going to be using 4 different psychiatric data sets for major depression, anxiety, alcohol use, and PTSD. Though I do want to note that I'm simply using these for the purposes of the practical, and in fact, all of these disorders have more recent and larger GWAS available than the ones I'm showing here. So just a side note that don't take this to be an indication of what is the best available data out there. I'm instead using these because it allows for comparing to results obtained on the GitHub, because we use these same data sets for those examples. A couple of things to keep in mind before getting started, reflecting common pitfalls that I see for people that are first starting to use the package. The first 2 relate to matching ancestry across all the different data sets that you're using. So genomic-SEM is not currently a multi-ancestry method, and the GWAS data that you're providing need to be within a single ancestry group, and that ancestry group needs to be matched across the included data. Similarly, the LD scores that are used to estimate LD score regression need to match the ancestry group of your included GWAS. GWAS data is now becoming well-powered and available outside of just European ancestry. So I would encourage people to think about running genomic-SEM models in other ancestries. But again, logistically, you need to make sure that it is within a single ancestry group in order for the statistical properties of LD score regression to work. With respect to power in point 3, a very rough approximation would be to only include GWAS data with a sample size greater than 10,000. But ultimately, GWAS power is going to reflect a lot of things outside of sample size, such as the heritability of the trait and the quality of the measurement. The more direct power consideration recommended by the original LDSC developers is to only include traits with a SNP-based heritability z-statistic reflecting the SNP-based heritability estimate over its standard error greater than 4. The reason for this being that they find that it produces interpretable estimates of genetic covariance, which is ultimately what genomic-SEM models. The fourth piece is actually something that you don't need to worry about, and that is the levels of participant sample overlap. LDSC directly backs out from the data the levels of sampling dependencies that will arise from participant overlap. So if you have 2 GWAS where you know there are some overlapping cohorts, but you don't know exactly how many participant samples overlap across the 2 GWAS, that's not a problem. You don't need to know it, and in the extreme case, you can even include traits that are non-overlapping or reflect entirely independent participant samples. The fifth point is just to make sure that you're not using a pruned list of, say, just the GWAS hits. you typically want to see at least 1 million rows in that GWAS data set that you're providing as input to munge. And then finally, the sixth point is probably the trickiest, which is how to get the correct sample size to provide to munge. So sample size is directly linked to the heritability estimate that you get, and it's easy to put in the wrong sample size. One reason for this is that the publicly available GWAS data that you download may be a smaller subset than what is described in the published paper. So for certain common data sets like 23andMe, they do not allow their data to be published publicly. And so the data set that you're using may be much smaller than what is being described in the paper. And if you include the total sample size from the paper, including 23andMe, you'll ultimately get a very biased estimate of the heritability. The second point is that for binary traits, this sample size needs to reflect the sum of its effective sample size, which relates to this point about the liability scale correction that I want to take a moment to describe now. So here what I'm showing is the continuous distribution of risk that we know exists for any human complex trait, where for binary traits, we also know that we have arbitrarily created some cutoff on this liability distribution, where we say that despite the continuous risk, at this particular point is where we would diagnose someone with the disease. And so this threshold T has a certain height in that standard normal probability density function, which we can use to actually perform a correction that I show in the next slide here. So here what I'm showing is the liability scale heritability on the left-hand side of the equation, which is calculated from the observed scale heritability that you get from LDSC, and then includes two additional parts. The first is that correction for the fact that we have arbitrarily truncated a continuous distribution of risk. And so here we have the population prevalence on the numerator and the height of the standard normal probability density function in the denominator here. If I'm losing some of you with some of this technical jargon about the liability scale correction, don't worry too much. The function is going to perform this corrections for you. What I really want you to take away from this is how you need to supply the information correctly for genomic SEM, specifically the munge function, to be able to perform this correction. The second part of the correction is for participant ascertainment. So this reflects the fact that when we are studying a disorder of interest, we are often oversampling for that disorder in our data set. If we're interested in depression, we put out flyers specifically recruiting for depression cases to come into our study, and so we create this distribution that is very skewed, which you can see in this plot here. So this is from a 2011 paper discussing the liability scale distribution, where instead of showing the liability scale bell curve that we saw in the previous slides, what this is showing is a distribution of phenotypic values in our actual data set that we've collected. Where here we have the controls, and then we have this very skewed distribution of cases because we've oversampled. So what they write in this 2011 paper is that in case control studies, the proportion of cases is usually much larger than the prevalence in the population. Yet estimates of genetic variation are most interpretable if they are not biased by this ascertainment. So that's what we're doing in that second part of the equation. The problem is that most GWAS reflect meta-analyses of multiple cohorts, and each of those cohorts are going to have different levels of ascertainment. For example, the DECODE sample from Iceland may have only a handful of cases and hundreds of thousands of controls for different diseases that are being studied, as compared to a different data set that may have sampled cases at a level so that they have around 50% cases and 50% controls. The point being that there are lots of cohorts that contribute to each GWAS, and the levels of ascertainment can be drastically different for each of those cohorts. The issue is that if you calculate ascertainment using the total sample size of cases and controls, you'll get a different answer than if you calculate the ascertainment at the level of the individual cohorts, which is to say that the sum of the parts is not equal to the sum of the totals, and it's doing it at the cohort level that we've shown in simulation and empirical results gets you that unbiased estimate of the liability scale heritability. So here what I'm showing is that same equation, but with the update to show here that we're looking at the sample prevalence V with this sub K now in the equation, where we're looking at the specific level of ascertainment within each cohort K and summing across those to get an accurate ascertainment correction within this larger liability scale correction equation. In practice, what munge wants you to provide is the sum of effective sample size. It's doing the same thing as doing the ascertainment correction, where it's calculating the effective sample size that you would have gotten had you had 50% cases and controls so that you can sum them together across the contributing cohorts. A reason for doing this in part reflects the fact that many GWAS softwares like Ricopilli, which is used by the PGC, will automatically include some of effective sample size in the GWAS output so that munge can just use that column in order to do this correction. And it's also something that you can calculate from cohort level information, which will often be deep in the supplementary tables or materials. And finally, in the Plan C option, you can directly back out some of effective sample size from the data. And we're going to look at an example of that for MDD in just a minute in the code. Before that, the last thing I wanted to go over in the slides is that the munge function takes 4 key arguments. The first is the name of the GWAS files, and you want to make sure that these are in the current working directory that you're working within so that the munge function can find them. The second is a hatmap3 file. So this is a reference file that is used to align to the same effect allele across GWAS, and also to restrict to a well-imputed set of 1.1 million SNPs that we have LD scores for in the second step 2 LDSC function that I'm going to go over in the next video. Some people have reached out expressing concern because they may have, say, 10 million SNPs in their GWAS file before running munge, and then it gets subset down to this much smaller subset of 1.1 million SNPs. But that's not something to be concerned about. That's as intended and based on the observation that 1.1 million SNPs is more than enough to get a good estimate of the LD score regression equation. The third argument is the trait names, so how you want those traits to be named in the files that are output from munge. And finally, the fourth argument is that tricky one, which is the sample size associated with the traits. And the last thing I want to say before turning to the code is I think in some ways munge, when done carefully and appropriately, does not need to be incredibly time-consuming, but can be the most time-consuming step. And if you will take the time and care to just carefully go through your traits at the beginning, you'll ultimately save yourself a lot of headaches in the long run by just making sure that you're working with cleaned, appropriate data at the outset. So let's go now to our R code here. So step 0 obviously is to load in the genomic SEM package. And then I'm going to set the working directory to this munge subfolder. And here what I'm going to show is how I back out the effective sample size, the sum of effective sample size for major depression, prior to even running the munge function. So this MDD file includes those 5 key pieces of information, A1, A2, RSID, p-value, and signed effect column, but we cannot calculate sum of effective sample size from the available information, and so we're going to back it out from the data. I'm going to go through this a little quickly, but details about the equation here are provided in a publication linked here in Biological Psychiatry, and also detailed on our GitHub. But to start, we're going to read in the MDD file, and then we need the minor allele frequency in order to back out the sum of effective sample size. The MDD file includes minor or allele frequency, and we're just going to make sure that we're calculating the minor allele frequency using this code here. We're going to remove the frequency column now since that's redundant, and then we're going to use this equation of 4 over 2 times MAF or 1 minus MAF, also known as 2PQ, times the squared standard error of the effect. Again, I haven't provided a lot of details about this equation for the sake of time, but I encourage you to look at this publication or the GitHub for more details. The point being is that there's a plan A, B, and C for getting that sum of effective sample size for any binary GWAS data that you might include. When you're backing out sum of effective sample size, sometimes it can produce out-of-bounds estimates. So we also calculate total effective sample size to calculate the backed out sum of effective sample size at 1.1 times the total NF and a lower bound of 50% times the total MF. And then finally, we output the data so that we can then provide this to munge. Again, MDD included all the necessary information in order to run munge, but we were not able to calculate the sum of effective sample size from available information. So we're backing about using that equation to then be able to input this to munge here in this next step. So here we've got that files argument, the hatmap3 argument, which is a SNP list within this LD scores subfolder. Again, this can be downloaded on the Genomics M GitHub. The name of the trait, we're supplying NA for the sample size column because I just showed how we backed out sample size, so it's directly in the data. Munge needs to find sample size somewhere. It needs to either be supplied to this argument directly, or it needs to be in the GWAS data that you're feeding to it. In this case, it's in this data set already, so we supply NA to this argument. And then we run munge. Just to note, while this is running here for the purposes of this example, I'm only showing MDD, but in practice, each of these could be a vector of multiple files, and for most of the examples, it in fact needs to be a vector of all the traits that you're including, but for munge, this is happening for each trait independently. As it's running, it's going to start printing how it's interpreting certain columns to screen. As part of doing your due diligence, you really want to make sure to inspect this output to make sure everything is working correctly. If you forget to look when you're in R running your code, you close it and you no longer have this in your screen. The good news is that if you go to the folder where you were running it, there's going to be a log file that permanently stores this information. So you can go here to see mark NA was interpreted as SNP, A1 is A2, it found the p-value, it interpreted the NF column that we created as the sample size column. And while there were 8 million rows to begin with, there were about 1.1 million rows left at the end. So again, something that you should inspect at some point, and if in the moment you forget to do it in R, you can go to that log file. So in the next video, I'll go over how to use LD score regression using these munged files as input, where what we've created by running munge is this .sumstats.gz file for depression, which is the formatted, cleaned, and quality controlled data that we're going to supply to LD score regression.